the interns. And finally, just thank you to the interns themselves. You know, it's always exciting to have a group of students come in, a pulse of energy and excitement and enthusiasm come into our center every summer. And you guys definitely brought it. You had dedication and enthusiasm, even in this sort of strange setting. This whole summer was mostly online. So, you know, I'm grateful for that. And you also sort of demonstrated this growth mindset that we talked about early on, that you brought your skills and experience to the table, but you were still here to learn, and that was evident every week. And it was fun to see you progress. So thank you so much, and I hope you enjoy the presentations from these budding water resource professionals.
And with that, I am pleased to announce that Olivia will be going first. Olivia is a rising senior. She is studying chemistry and environmental science in the College of Arts and Sciences. As she gets her screen ready, she will be presenting on manganese in the local Ithaca area. Olivia, whenever you are ready.
Awesome. Thank you so much for that introduction, and thank you all for being here. I will share my screen. Whoever's the host, would you mind letting me screen share? Currently says host disabled. Participant screen share.
You should be all set.
Thank you. Awesome. I have access to it now. Can you all see this?
Yes, we can.
Amazing. So I will hit present. All right. So the project that I worked on is analyzing manganese in the Ithaca Reservoir sediments, and I worked on this with another intern, Ashley Sizzle, under the mentorship of Dr. Matthew Reed. So the motivation behind this research project is that the Ithaca Reservoir is an important drinking water source for large parts of the Ithaca community. And in 2016, there was a drought during the summer that caused manganese levels to increase above 0.4 milligrams per liter. And above this level, the tap water turned brown, and it can be toxic to nervous system, kidneys, and reproductive functions. So the Ithaca water treatment plant wants to prevent this from occurring in the future. And one possible source of this manganese release into the reservoir is the sediments at the bottom of the reservoir. So the specific project that I've been working on is analyzing the impact of the depth of the sediment and the location within the reservoir on the amount of manganese released by reservoir sediments. So one question is if the older, deeper sediment is still releasing manganese, or if it's largely being released by the younger, fresher sediments. And also have been comparing two methods of measuring manganese release from the sediments, which I will get more into later. So the first step is sediment collection. So initially, I canoed out onto the reservoir with the help of some Ithaca College folks, and usually with an oar, but sometimes with a snow shovel, as you can see in this picture. And then sticking a long sediment core into the bottom of the reservoir. And once it's pulled up, the sediment will be extracted. And this picture on the right is the sediment, which I cut into 5-centimeter sections. And at the top here is the shallower, newer sediment, newly laid sediment. And then at this bottom here is the deeper, older sediment. So this is an image of the Ithaca Reservoir, the parts that we sampled from. Here at the top is a dam. And so previously in 2019, there were 5 locations that were sampled, one being closest to the dam to kind of through to the middle of the reservoir, 1 through 5. So I used these for collections. And then also this summer we went out in June and got these 3 locations here in reddish orange that I also used to analyze later on in the summer. So 2 methods that I used to test the release of manganese from the sediments. The first one on the left is the sediment incubation method. So I took these sections of sediment cores and put them into glass vials on the left here with synthetic reservoir water at the top, including these compounds. And it did extractions from these vials from days 1 through 20 with needles. And then another method that I tested is the CBD method, when CBD stands for sodium citrate, bicarbonate, and dithionite. And so I added this to the reservoir sediment, heat it up in a water bath, this image on the left, and then shake it on a vortex, this image on the right, to kind of vigorously release the total reactive manganese. And for both methods, used the inductively coupled plasma mass spectrometry, which is ICP-MS for short. And this machine analyzes the manganese concentration released into the water above the sediment. So this is some data from the sediment incubations of one single core wanted to do a trial run of the method to see what results we would get. And so this is a core from 2019 at 2 different depths. So this depicts in blue the shallower depth and in orange the deeper depth over the span of 14 days. And then it's showing the manganese concentration in milligrams per liter. And this graph is kind of revealing that more manganese was released from the shallower, younger sediment from this trial run of a single core in 2019. So based on this, I wanted to do a core at more than 2 depths. So this is a sediment core that we collected this past summer in June at 4 different depths. So at a shallowest depth of 0 to 5 centimeters, and then 10 to 15 centimeters, 20 to 25 centimeters, and then the deepest depth of 30 to 35 centimeters. Also over the course of about 20 days, measuring the manganese concentration released into the water. Overall, the trend shows that the shallowest depths, the most recent sediment, was releasing more manganese than the deeper, older sediments. And then another thing that we wanted to analyze was the spatial variation of manganese release across the reservoir. And so I took 3 different sediment cores from 2019 at 2 different depths. So the ones in black here nearest to the dam, and then the ones in orange kind of in the middle of the reservoir, and then the ones in blue farther from the dam. And all of the circles represent the shallower depth, and then all of the lines represent the deeper depth. So as you can see, there aren't very many clear spatial trends in this data, which we wouldn't necessarily expect because we're seeing more manganese within the sediments nearer to the dam. And so this could be because the sediments collected in 2019 were not stored anoxically. They were stored where oxygen could go in and out of the bags. So I wanted to, in the future, perform this test again on newer sediments from 2021 that were stored in sealed anoxic bags to hopefully get some more clear pictures of the data. And then this is some results from the sediment incubation method versus the CBD method. So I took 1 sediment core at 2 depths. So on the left is the shallower depth and on the right is the deeper depth, and compared the manganese released from the incubation method in blue compared to the CBD method in orange. As you can see, the CBD method in orange released a lot more manganese. And this could be because it's most likely because it's a more vigorous method of heating and shaking. And the incubation method is most likely to be reflective of the reservoir itself because the reservoir isn't being vigorously shaken and heated at all times. So in the future, I'll focus on the incubation method. So future directions and goals of this project. So I want to perform incubations for the newly collected sediment cores in 2021, 1 through 5, which will span the entire reservoir to get a location. And also for each core from 0 to 30 centimeters depth, so 6 sections. So I can really see the effects of the depth and the location across the reservoir. And also, this will also help the Ithaca water treatment plant decide where to dredge. So dredging is kind of collecting sediment from the bottom and pulling it up. And so if we are able to determine where the locations of the most manganese release, that will be the locations where the Ithaca water treatment will want to dredge the sediment. And also another goal is to scale these incubations up from just portions of the reservoir to the entire reservoir to calculate the total manganese that could possibly be released by the entire reservoir. And this will help the Ithaca water treatment plant be able to treat the manganese with oxidant dosing and be able to manage future manganese release because we want to overall prevent manganese levels from getting high in the drinking water and toxic to humans. Yeah. So I would like to thank a lot of people here who participated and helped with this project. People from the Water Resources Institute, Ecological Engineering Lab, Ithaca Water Treatment Plant, and then also Christopher Sinton's group from Ithaca College. Yeah, thank you so much. If there are any questions, I believe we have 5 minutes now to talk about them.
Yes, we do. If you want to use the reaction feature to raise your hand, I can call on you. While we wait for questions to come in, I know I have a personal question. Olivia, what environmental conditions or factors could affect the amount of manganese released by the sediments?
Yeah, that's an awesome question. Thank you for asking it. So actually using environmental factors as predictors for manganese release is what Ashley has been working on a lot this summer. So shout out to her. Some factors that do impact manganese release, higher temperatures can increase the activity and respiration of microbes in the reservoir, which causes oxygen depletions like anoxic conditions, which can reduce manganese 4, which is in solid manganese dioxide, to manganese 2, which is soluble and gets released into the reservoir water, which would increase manganese in the water. Along with the stagnation of water, this also increases microbial respiration. These two are some big reasons why summertime is typically the time where we see higher manganese release into the water and is more of a concern. And also the presence of organic ligands like humic acid or sodium lactate, those can bind to manganese and convert it into a solid form. So it will get removed from the water and stay within the sediments. And then another factor, it can be the pH of the reservoir water. And at higher pH values, some aqueous species like carbonate become more abundant, and they're more likely to bind to the aqueous manganese and also form these solid precipitates. I think a main one is like rhodochrosite, which is manganese carbonate, which also removes manganese from the water because it'll become a solid. So yeah, there are a lot of other factors, and Ashley's been working on kind of a predictive model, taking all of these environmental conditions and being able to predict the manganese that will be released. Yeah, thank you for that question.
So cool. If you are in a loud area, feel free to write questions in the chat box and I can ask them for you. Ingrid, you may ask your question.
Hi, Olivia. It's great to see how this project turned out. You did a great job. I was curious just more broadly, I know you're heading into your senior year at Cornell, so I was wondering, kind of having done this project and this internship, kind of how you're thinking about next steps in terms of what you want to do with engineering, if you want to continue on this, do other things. Yeah, just wondering kind of where you landed by the end of the summer.
Yeah, so this summer, I've really had a great experience doing research with Dr. Reed and other people in the lab and at the Water Resources Institute. I think in the future, I do want to continue with environmental engineering and water resources, and also kind of see what equity or sustainability lens if possible. Yeah, I think that I've just had really great experiences, and I do want to continue this specific research project. I think there's a lot more things that we can look at with the like environmental factors and like humic ligands and things like that. So I think there's a lot of work to be done with this project and also in the future. Yeah. Thanks for that question.
We have time for 2 more questions.
There's questions in the chat, T.
Oh, beautiful. All right, I have a question from Kathy. Do you know what the source of the manganese is? Naturally occurring or man-made? Start with that one.
Yeah, that's a great question. I know that manganese is a naturally occurring element and it's essential for a lot of life processes. So if we fully wiped out the manganese, that would be an issue for a lot of biological processes. I'm not entirely sure if specifically the like where the manganese running off into the reservoir is. I know there's there could another source besides the sediments could be surface water runoff. So there could be some factors there as well. And I see your second question, a theory as to why the newer sediment releases more sediment than older. So I think that, yeah, this is a good question. Likely some of the, like the older sediment will have released manganese into the newer sediment. So kind of will like be collecting more into the newer sediment. And then there's also this kind of like sediment water interface almost that any of the soluble manganese, if it ends up binding with other elements to form precipitates, they'll kind of collect at the surface. And any manganese that's coming from other locations into the reservoir, if when it settles, it'll kind of lay on top of the fresher sediment. So this is kind of a thing to consider when treating it or treating the water for manganese, is that the sedimentation rate. So kind of how quickly is newer sediment building on top of the older sediment. And we predict that it's laying on top of each other. The sedimentation rate is pretty fast, pretty rapid. So that kind of brings into question how, if the Ithaca water treatment plant does dredge, how long will it be effective for if this sediment is collecting at a fast rate? So, yeah, thank you for those questions.
So we are going to move on to our next presenter. Thank you so much for that, Olivia. That was awesome. So next up, we do have Joseph Nakua, a rising sophomore. He's getting his degree in biological engineering. Go ahead and take us away, Joseph.
Thank you very much, Ashley. Joseph is indeed trying to figure out how to share screen. I think he figured it out. There we go. All good? Okay. So I will start. Hello, everyone. I'm Joseph Nokwa. As aforementioned, I am a rising sophomore studying biological engineering, and I am going to present to you today what I did over the summer with the New York State Water Resources Institute alongside T, one of our amazing hosts over here, and under the mentorship and guidance of Dr. John Sinda, also present in the audience today. So, responding to risk from flood and COVID-19 beyond partisanship through experience. In 2020 and 2019, there have been 2 very major hazards that have dominated public attention: COVID-19 pandemic and climate change. The COVID-19 pandemic having brought the world to a total halting state. Both of these issues have had major ramifications in the state of New York. At the onset of the pandemic, New York State has suffered a tremendously debilitating surge with over 2 million cases and 50,000 deaths. Additionally, in recent years, due to the influence of climate change, New York has exposed itself to, well, that's redundant, to major climate change exposures, mainly revolving around the idea of flooding. Sea level rise increases flooding not only along the coastline, but also in inland areas, for example, those that are in contact with the Hudson River estuary, which as an estuary system makes it so there's a direct proportionality between sea level rise and riverside water level rise in communities such as the ones we're going to be talking about today. I will acknowledge that This could have not been possible without the Hudson River Estuary Program and the New York State Department of Environmental Conservation, whom helped the individuals from Global Development last year to conduct the surveys that I'm going to be talking about today, the data we analyzed over this summer. The hope is that by understanding better how COVID-19 risk and flood risk relate to each other, or how people can perceive these different risks and act towards them, we can better target policy measures in local government and better reach out to communities to make them more risk aware and allow them to better enact action. So Troy and Kingston, the study was indeed focused on Troy and Kingston, communities that have had recent flood experience. As of, for example, 2011, Troy underwent a very large flood influenced by the arrival of Hurricane Irene to the state of New York. The data compiled comes from 4 focus groups, which add up to a total of 32 participants and a 499-person questionnaire or household survey that was administered in both towns. Additionally, these 2 towns had already participated in projects alongside the Hudson River Estuary Program, so they made for ideal partners. Our research, we wanted to understand different things, how people assess risk and subsequently act on it. So what influences, for example, flood risk? Is there some form of politicization on these issues? What influences someone to take action towards mitigating risk? We wanted to see if flooding was politicized because of its potential intrinsic association with climate change. And of course, this was with the goal of helping policymakers and local government better understand how to reach communities regarding flooding. So before we continue, I will explain some of the mechanics of the questionnaire to better comprehend the results that I'm about to show to you. So whenever I mean protective measures, this was a list or a checklist that was given to individuals. In the case of flooding, composed of 15 measures that ranged from elevating your house using stilts to moving your documentation to a safe location in case your house were to flood. And in the case of COVID, it refers to a total of 4 actions ranging from wearing a mask to reducing your monthly outings in order to obtain essentials. We also asked individuals if they were Republicans, Democrats, or independents/others in order to better assess where on the political spectrum they fell. Additionally, flood experience was assessed via asking individuals if they had suffered directly from flooding in the past in the form of perhaps basement flooding, first floor flooding, or if they had suffered the secondary effects of flooding, for example, contamination in drinking water. For COVID-19, we used a known health condition or a known health risk that could potentially turn into a comorbidity if you were to contract COVID-19. Individuals were asked whether they had one or if they didn't, of course, to the best of their extent, as there are conditions that individuals could be unaware that they have. As we can clearly see with the figure on screen, our response to 2 prompts regarding concern of COVID and concern of flooding, generally speaking, there is a much lower concern for flooding and a much higher concern for COVID-19, which is actually something that we did indeed foresee, taking into account of the widespread alarm for COVID-19 over the past year and a half. Additional to that, the fact that the communities that were part of the study are Democratic leaning, urban, and racially diverse. We also wanted to assess views on climate change, as it is extremely important in order to better understand how people perceive flood risk. The views on climate change have changed over the past decades, especially with a particular demographic regarding more conservative leaning individuals. There has been a shift in not believing in climate change to indeed believing in climate change. However, there still remains some form of political party polarization. As can be seen on screen, Democrats and independents largely believe climate change to be human-induced or anthropogenic in origin, and Republicans, even though believing in climate change, on average, believed climate change to be a natural occurrence. And we believe this to be important because given how climate change impacts flooding directly, we would have expected some level of similar polarization revolving around flooding. However, we were wrong. As can be seen on screen, we can see that we did get the expected polarization around COVID-19, where Democrats were most likely to be concerned and Republicans were least likely to be concerned. However, across party lines or across party identifications, there is no such polarization for flooding, which fundamentally contradicts what we believed originally. Looking at protective measures taken based on party identification, we do see very much diverging patterns, specifically between people who have not had previous flooding experience and people who have had previous flooding experience. People who have not had previous flooding experiences averaged roughly 3 out of the 15 actions that I mentioned earlier. and people who did have flooding experience averaged a total of 6, which is essentially almost double. And even though polarization is not evident, there is a a statistically considerable difference between Republicans and independents without flood experience and those that do have flood experience. As for COVID-19, partisan differences were again evident. Amongst individuals who do not have a known risk condition, Republicans took significantly less actions than Democrats and independents. However, having a known risk condition managed to certainly offset this difference, or in a sense, eliminate the partisan leanings or partisan trends that we saw for COVID-19 concern. Republicans who reported having one or more risk condition were more prone to taking action, and they were actually similarly prone to taking a similar amount of actions to individuals that identified as Democrat that also had risk conditions. So what does this all add up to? Indeed, political polarization in the US is something tangible and unavoidable that will implicitly affect how we can deal with communities in terms of assessing them on risk or making them risk aware. So, however, with the results that were obtained from the survey, we can indeed catch some glimmer of hope as to what we could potentially do. Ashley, are you raising your hand?
You got 2 minutes left.
Oh, okay, my bad. Oops. So first, indeed, while climate change polarization does exist, it does not translate into polarized responses on flooding. It seems as you do not have to recognize flooding as a direct result of climate change in order to better adapt to it, which is good for flood managers and risk managers and local governments, because focusing on the concrete matter of reducing flood exposure and vulnerability could lead towards building a more flood resilient community. Additionally, experiencing danger or loss can offset partisan leanings. For flooding, as aforementioned, past experiences are the strongest predictor for both concern and action. And for COVID-19, knowing that one is at a higher risk of suffering from illness if one were to contract the virus reduces the gap between political party lines, effect that would generally reduce protective measures. So this does bring a fundamental issue, and it is that we cannot part from the premise that individuals have experience with certain type of natural phenomena or that they have some form of risk condition. So that leads us to the fundamental question. What can we better do for individuals to understand the risk that they could be potentially exposed to if they have never seen it in the past? And research suggests, and also the results of the survey suggest, that perhaps telling the stories of people who have undergone these challenges in the past may be the way to enact people to take protective measures. And even though I do want to acknowledge that this is not perfectly translatable into work in the rest of the United States of America, it does give us some form of hope that it could work out with communities that are similar in composition to Kingston and Troy. So perhaps liberal, racially diverse urban communities that are near the Hudson River. I also do want to leave with a general conclusion, and it is that despite political party affiliation and fundamental differences on how we perceive risk, as empathetic beings, we are joined by the human experience, as I like to call it. Experience is our most effective predictor for protective action, and perhaps by telling the stories of those individuals that we care about that have undergone these challenges, we might just be able to reach beyond partisan lines. Thank you very much. If anyone has any questions, I'll welcome them gladly. If I can't answer them, I'll shoot you an email and be sure to answer them within 24 hours, business hours, by the way. So any questions? Yes, T.
Wow, thank you so much for such a thorough and detailed report of this. I gotta ask, what are the potential directions this project could take in the near future?
Perfect. So you know about this. T has been working on fabricating outreach material and flood awareness material for the Hudson River Estuary Program, which has been sent in for review today. So the hope is that it will be ready and it will be usable soon. And in terms of what I just presented to you, it is largely drawn from a policy brief that will hopefully be published just ready by the end of August and published soon after that, focusing on contrasting COVID-19 concern and flood action and risk in Kingston and Troy. Yes, Rewa, my bad.
No, that's good. Great work. I was just asking, I wanted to ask if, like the work that you did about flood risk perception, how much of how communities perceive risk does it have to do with how risk is communicated in terms of like terms that we use, such as the 100-year flood? And these terms have been problematic and criticized for not accurately representing risk. So you can't always blame communities for not understanding risk well enough. And I was wondering if that came up in your literature review at all and whether you thought about that.
Thank you very much for the question. Indeed, it did come up in the literature review in repeated occasions. What I mentioned at the beginning, the lack of flood awareness that communities had, is indeed very heavily influenced by the terminology that federal agencies have been utilizing to refer to flood terms. For example, what I think is the most misleading one possible is the 100-year flood, which leads individuals to believe that if I had a 100-year flood in 2011, the next one's coming in 2111, therefore I can just bive and ignore it because I will most likely not live another 100 years. However, of course, we know that it does not mean that. It just means that there's a 1% probability every single year compounded for 100 years. There's a 100% probability of the flood occurring. But yes, it very heavily influences how communities react to risk in terms of flooding. So that is why we're helping them potentially have better wording or better phrasing, to better reach out to the rest of the community and better have them mitigate flood, or that's the hope, of course. Did that answer the question? Okay.
I think Kathy has the last question in the chat.
Oh, I did not see it.
Oh, no, that's okay. It sort of just got answered. I was concerned with you had made a statement, I believe, in the beginning that not many people were aware of flooding. And I just wanted to know what that was based on. So it sounds like there's a difference in my mind of people being aware that flooding happens and then understanding the flood risk. And then my question was, do you think that awareness is based on where they are? If they're in a community that really hasn't had a lot of flooding, is it just something that they don't really think about? So they're not out there thinking about it and going, what should we do? And climate change isn't impacting me because my house isn't flooding.
Thank you very much for the question. I do think it is partially influenced based on the location in which you're at, especially if you're located within a special flood hazard area as denominated by FEMA or not. But I also do think that it is very important where you are time-wise, where you are located time-wise. Have you undergone a flooding event in the past? Has your city undergone a flooding event in recent times? But yes, most definitely place is extremely influential on people's chances of being flood aware, but also how their government deals with flood outreach, and whether there's been recent events, I think might be heavier in terms of influence on flooding awareness on a community.
Thank you. Great presentation, by the way.
Thank you.
All right, with that, I will have Joseph close your window so that we can introduce Noelle D'Antonio. She is a rising junior as well as a rising superstar studying environmental engineering in the School of Engineering. Today, she will be presenting not one, but 2 presentations for both of her projects. Noelle, take it away.
Thank you, T, for that introduction. For my first project, I will be presenting on harmful algal blooms. And I have done this work along with Joseph Nokwa, a fellow intern, Professor Ruth Richardson, and Nan Wong, both a part of the Cornell School of Civil and Environmental Engineering. And if you're wondering what this picture is in the background, it's a picture of a bloom under fluorescent light under the microscope, and we'll talk about that a little bit more later. So a little introduction to harmful algal blooms. There are colonies of algae that have grown to be much larger than normal, and they're typically caused by eutrophication and warm weather. And they're harmful because they can lower dissolved oxygen levels, and they also produce toxins that are harmful to humans and wildlife. And so we need to know if there are these toxins in the water caused by these algal blooms. So our project is focused on Cayuga Lake, which is currently in a HABs crisis. The main HAB genus of concern in Cayuga is Microcystis, and it produces the liver toxin microcystin. On the right here, you can see a Microcystis colony. And it's from a bloom on June 21st. And that is kind of problematic because it is earlier than what we would expect for algal blooms. And traditional methods for determining the levels of microcystin are timely and costly. One method called ELISA, it costs $150 or more, and it takes longer than a day to process. And if you want to go in the water, it would be really nice to just know right away what the levels of microcystin or other toxins are in the water. So we want to create a fast, easy, and cheap method to determine whether water has elevated levels of microcystin. And we will do this using community scientists who take water samples and collect pictures with a microscope and standardized backgrounds. As you can see on the right, this is one of the pictures taken with the microscopes. And we also use image analysis software, free software, ImageJ, and it'll help. And we hope to be able to predict toxin status with it. Right now, we can only go about as far as chlorophyll A levels. As you can see in this graph, the bloom density is the number that we get out of the image analysis software. And then the chlorophyll A level is what we calculate with bloom density. We're able to get an R² value of 0.5454. Unfortunately, we're doing this using some human assistance, like they have to look at the image analysis and determine whether the image analysis has done a good job, and then we use it to calculate chlorophyll A levels. So we're not exactly where we would like to be, but we hope to improve. So our project has a lot of moving parts. So I'm just going to talk a little bit about how it all fits together. So first, the volunteers, they collect the water samples. And then on the left, the volunteers take pictures of samples with a microscope. These pictures are then uploaded to Google Drive. And then someone at our lab will analyze the image with ImageJ software. And then from there, the chlorophyll A and bloom density levels are estimated. Now on the other side, the volunteers also deliver the samples to the Community Science Institute, or CSI for short. CSI tests the toxin levels and other properties of bloom. They test them officially. And then our lab also picks up the water samples from CSI. And we will look at the samples under the fluorescent microscope. We also filter the samples and then extract DNA, which is used for other research. We do outreach to engage volunteers. We have bi-weekly meetings where volunteers can come and ask questions. And then we also have videos and other resources that outline the process from taking the water samples to using the microscopes to uploading pictures onto the drive. Right here is a video that Joseph and I made. I'm just using the microscope. It's like a less expensive microscope, so we can distribute them to the volunteers. You can see it just shows the picture on the phone screen, and you can take videos and pictures. In addition to outreach, we have also done a fair amount of lab work. So we do fluorescence microscopy, which I mentioned before. Fluorescence microscopy, it allows you to see some parts of the bloom that are less visible under normal light. And this is because photosynthetic organisms naturally glow under fluorescent light, as opposed to non-photosynthetic organisms, which generally do not glow under fluorescent light. So you can see pictures. On the left here is a picture, just regular microscopy, and then on the right is fluorescence microscopy, and you can kind of see how there's different things that you see under the fluorescent light. In addition to the microscopy, we filter samples, and the filter traps DNA, which is then used for research, as I mentioned before. This right here is a picture of a typical filtering apparatus. So then for the future of the project, we would like to update the ImageJ analysis so that it can estimate microcystin levels in a bloom. And we'll do this either using some sort of human assistance to tell to tell the program if there's a certain type of algae in the bloom, or also we would like the software to be able to know whether there is microcystin in the bloom, because there are different types of algae. And we'd also like to create an interface between the remote picture taker and the ImageJ software. We do this, we're thinking possible app development or perhaps a web page, because right now the volunteer uploads the pictures to the drive and then someone from the lab has to run it through the software, and we would like to just streamline that whole process. And then we would also like to expand the project to more lakes. This summer, we've expanded to Chautauqua, Owasco, and Honeoye, and we'd like to go further and involve more lakes in this project. Now, to finish up, I would just like to make some acknowledgements. First, to the Richardson Lab Group, Professor Ruth Richardson and Ann Wong especially, to the Community Science Institute and the Hab Harriers volunteers. The volunteers make this all happen. And also the Finger Lakes Institute and the New York State Water Resources Institute. So that is it for my first presentation. I think we can take a few minutes of questions now, and then I'll get into my next presentation.
Hi, can I ask a question?
Yes.
Really fascinating project. Thank you, Noel, and the other person that worked on it. So I had a couple of questions. Is your chlorophyll concentration from a fluoroprobe, or is it extracted in the lab?
So the chlorophyll concentration that we're calculating that was on that graph, this was from the image analysis software. The Community Science Institute tests the chlorophyll concentration as well, but I don't know exactly how they do it.
So on the graph that you showed, was that just calculated from the ImageJ, or was that
yes,
an independent analytical measure?
That was from ImageJ.
Just from ImageJ, okay, thank you. And can you explain why the microscope images have those big black stripes going across them?
Oh yes, that is, so we can tell the size of it. We have like a micrometer that we use as the background. I believe it's 500 microns in between each line. So just so we can tell the size of the blooms.
Okay, thank you. And one last question and then a comment. So how do you account for the fact that some blooms can start off non-toxic and then become toxic, so that their toxin concentrations are not correlated with their chlorophyll concentrations?
Yeah, so that is what we're working on right now, because well, in Cayuga Lake, we typically have dolichospermum, which does not really produce toxins, and then microcystis, which does produce toxins. So that's one of the reasons why we're not able to detect microcystin levels right now from the image analysis software. What we would like to do is program the image analysis so that it can be able to tell, like, if this is dolichospermum, so it's not producing toxins, or this is microcystis, so it is producing toxins. And then, so hopefully that'll be able to account for it.
Okay, brilliant. Thank you very much for your answers to the questions. I just wanted to make sure that everybody is aware of the DEC guidance on harmful algal blooms. And it's based on the fact that you can't tell how toxic a bloom is based on what it looks like, at least with our eyes. So the DEC guidance is see it, know it, avoid it. Okay, thanks.
Thank you.
With that, we should probably move on to the next presentation, Noelle.
Yes. So this is my next presentation. It's about biological acoustics in the Hudson Estuary. And I've been working with Maya Nimisto as my mentor. Now this project, there will be some audio in it. So let me be sure that everyone can hear. If you guys could just give me a thumbs up. Are you guys able to hear?
Yes, we were.
All right, great, thank you. So maybe wondering, why is the study of bioacoustics important, or perhaps what is the study of bioacoustics? So bioacoustics is a study of the sounds of nature, and it's really important because you can learn a lot about what is going on with the animals from the sounds that they make. They make sounds for mating, for marking territory, and for communication, and other things as well. We can listen to ecosystems and analyze what is going on. And in addition, it's also important for us to know how we as humans may be changing the environment. We put out a lot of noise pollution, and it's something that is not often thought about. So a brief introduction to my project. So our program as a whole, like in the larger research group, our goals are to record different areas of the Hudson Estuary using passive acoustic monitoring. Passive acoustic monitoring being just putting a microphone and a hydrophone out in the wild and letting it stay there, and then listening to whatever comes. We just keep it there for a while, and then we retrieve it and analyze that data. Then we'd also like to create a library of the fish sounds in the Hudson Estuary that would allow us to interpret the recordings. Now, my project within this is a little different. It's about outreach. So I am trying to create an educational tool that will be displayed at the NYSWRI Point Environmental Center. And this is just an educational center. And the goal of my presentation or exhibit will be to educate students about bioacoustics in general. Now, before we can listen to the recordings, we have to put the microphones and hydrophones out. So here's a picture of my mentor placing the hydrophone in the water, and then up here in the tree is where we would have the microphone. And this is at Black Creek, and we will talk a little bit more about Black Creek later. Additionally, some creatures are taken back to the lab. We did this by seine fishing, which is dragging a net through the water. You can see right here the net. We go against the current, so the fish swim right into it. And then when you lift up the net, you can retrieve the fish. And then we take them back to the lab and we put them in a tank, add a hydrophone, and then we can kind of listen to what they're saying a little bit better than if we just had a hydrophone out there in the creek. And on the right here is a crab that we got from seine fishing as well. We took that back to the lab to listen to. Additionally, so my project is concerning the Hudson River estuary. So I just thought I would show you a map of that. It is outlined in red here. This blue is the Hudson, and the sites that I will show you will be right here, just north of Poughkeepsie. So right now I'm going to show you some examples of the educational tool that I'm creating. This first slide, it will be from the NYSWRI Point Environmental Center, and here's where it is on the map. It's right there on the Hudson. So we'll be focusing on anthropogenic noise, but first I would like to introduce the spectrogram view. It's kind of a visualization of the audio. So on the x-axis here, you have time. On the y-axis, you have frequency. So anything that's higher up is higher pitched. And then the color just shows what is louder. So if something has a brighter color, then it is louder than something that is not as bright. So we will listen to this recording. And as you can tell, anthropogenic noise is quite loud. It's louder than the biological sounds. This can be problematic because multiple studies have shown that animals may change their behavior in order to compete with the sounds. They may start calling at different times, or they may modify their calls so that they can be heard over this anthropogenic noise. So for our next site, we're going to Black Creek, which is, you saw pictures of before. Is right here across the Hudson from NYSWRI Point. Here we'll be listening to some underwater sounds. Unfortunately, we can't really identify these sounds yet because very little is known about them, but hopefully after my program does more research, we will be able to identify these sounds. So here for this first one, you're going to be listening for a croaking sound. And then for this next sound, you'll listen for something that sounds like a heartbeat. These were both taken from underwater at Black Creek. It can be kind of difficult sometimes to use passive acoustic monitoring underwater just because the water itself can be really loud, so you can't really hear the fish as well. So just some key takeaways from my project. Biological acoustics is a really valuable ecological research tool. Soundscapes, they provide important information about ecosystems, and then we can use this information in conservation efforts. Additionally, this research can be used in outreach to excite the public about soundscapes, let them learn more about biodiversity, because sometimes you can't see animals, but you can hear them. And then teach them about underwater soundscapes as well. So to finish up, I'd just like to make some acknowledgements. New York State Water Resources Institute, Cornell Lab of Ornithology, New York State Department of Environmental Conservation, the Hudson River Estuary Program, Alex Flecker, Aaron Rice, Maya Nemisto, and Patrick Baker. So thank you. Are there any questions?
Thank you so much, Noelle.
Hey, Noelle, this is, I don't, can anybody hear me?
Yes.
Hi there, this is Chris Bowser. Noelle, we met at the NYSWRI Environmental Center. How are you?
I am great. It's nice to hear from you.
Oh, that's terrific. I'm sorry to be only attending by phone today. Thank you so much for the work that you've done and the time and commitment that you put into this. And I want to make a special thank you for you and the rest of the crew that came out to NYSWRI Point and not only did some field work, but also had a really great sit-down brainstorming session about things. And Noelle, you and others, but you in particular, really helped me understand sort of, or at least helped me explore some of the potential educational outreach and applied research for this particular bioacoustics project. And I was wondering, since, you know, that was about a month ago, and I'm sure that there's been a lot of thoughts ruminated in your head. So when you think about the use of this for an environmental center for outreach and applied and education, what are some of the things that excite you the most in terms of next steps that we should be thinking about?
Oh, that is a great question. So I think what excites me the most is the thought of having like a multi-sensory experience, because I think with most places, exhibits, you just have something that's visual. With this, you also hear it as well. And I feel like hearing it will really allow people to like better understand the environment and develop a greater appreciation for it.
Awesome, Noelle, and I hope that we can continue to entice you to work on this project with us. So thank you very, very much.
Thank you. Yes.
Awesome. Well, thank you, Noelle. We are going to have to move on to our next presenter. Awesome presentation. So our next person up is Julia Gonzalez. She is an environmental engineering major and also a rising junior. Go ahead, Julia, take us away.
Thank you for that introduction. One second while I share my screen. Can everyone see? Great. So thank you for that introduction again. My name is Julia Gonzalez, and I will be presenting on PFAS in New York State. I worked with Olivia on this project, and we were mentored by Rasil, so thank you to both of them. So to start, what are PFAS? PFAS stands for per and polyfluoroalkyl substances. They're classified over 4,000 chemicals, and they're found in many common products like Teflon, microwave popcorn bags, and other food packaging, aqueous firefighting foam, industrial plastics, and they're resistant to degradation in fire and have some other interesting chemical properties that make them valuable for a variety of applications. However, they're also possibly linked to a variety of diseases, including kidney cancer, testicular cancer, ulcerative colitis, birth defects, pregnancy-related hypertension, high cholesterol. We only started public research on these chemicals about 20 years ago after some major lawsuits against the manufacturers of some of them. So we don't have a full understanding yet of the possible health impacts that they can have on humans. And we also don't yet have a federal legal limit on the amount of PFAS you can have in the environment. We do have an EPA health advisory, which is an unenforceable recommendation of 70 parts per trillion in water for two of the more common PFAS, PFOA, and PFOS. And in New York State, we do have a legal limit or a maximum contaminant level of 10 parts per trillion each for PFOA and PFOS. Now, with PFAS being so possibly dangerous and being so widespread, people want to know where we're actually detecting them. And last year, 2 interns from NYSWRI, Harmon and Carolyn, made this map of PFAS detections in Long Island. And this year, we wanted to add on to that research and map detections in Rensselaer and Orange counties. I'm going to get more into this later, but we chose these counties because they're home to some major drinking water contamination issues. Towns, villages, and cities in these counties have had their drinking water supply shut off because of high levels of PFAS contamination, so we thought these were good places to focus on. So the goals of this project were mapping detections of PFAS in these counties, looking at some remediation measures, comparing some demographic and socioeconomic data, and then developing a story map that combines this year's and last year's research. And these are just some of our data sources. I'm not going to get too much into each of these individually, but please feel free to ask me about them later. The main takeaway here is that there's not yet a comprehensive monitoring program. We do expect more monitoring to happen with the New York State MCL coming out in late 2020, so that data will start coming out soon. And another takeaway here, we mostly map the prevalence of PFAS, so we map places where there are detections and not so much places that have been tested but don't have any detects. So getting into the maps. This is our map of Rensselaer County. If you click on any of these points, you get a little pop-up that gives you some information. One key point is the sum of the available PFAS concentrations. Now, not all points test for the same number of PFAS, but this is sort of a general measure of how much PFAS we know is in a specific location. And that's how the map is organized, as you can see in the legend on the side. So looking at this map, you might notice that a lot of these points are clustered here in the northwest corner. And I want to get a little bit more into some of these clusters. So first, the town of Petersburg. The town of Petersburg was home to a major drinking water contamination issue. A local plastics manufacturer, Taconic Plastics, used PFAS in some of their manufacturing processes. It ended up entering the drinking water supply. Residents were forced to switch from their well water to bottled water, and they're currently in litigation suing Taconic Plastics for their alleged role in the contamination. On site at Taconic Plastics, PFOA concentrations were as high as 150,000 parts per trillion. And at a landfill where they disposed of some of their waste, PFOA concentrations were as high as 2,000 parts per trillion. Another site in Rensselaer County I want to talk about is Hoosick Falls. Like Petersburg, they were home to a major drinking water crisis. Residents actually identified that they had a contamination issue after paying to get their own water tested because they noticed high levels of cancer among themselves. Like Petersburg, they also had a plastics manufacturer located here called St. Cobain that used PFAS in some of their manufacturing processes, and it ended up contaminating both the municipal drinking water supply and some private wells. On site at St. Cobain, PFOA concentrations were as high as 130,000 parts per trillion, and this facility was in a generally residential neighborhood. And one point I do want to call out is their Little League complex, where PFOA concentrations were as high as 280 parts per trillion. This was a groundwater sample, so it's not like there were children splashing around in PFOA water, but in the context, it is a bit interesting. And one other point I want to call out in Rensselaer County is the Tomahawk Reservoir. It provides water to the cities of Troy and Rensselaer, which which just for reference make up about 37% of the population of the county, as well as some other towns and villages, and PFAS was detected there. It is within New York State legal limits, but PFAS was detected there, and this reservoir is also used as a fishing ground, and you can be exposed to PFAS through ingested seafood. Moving on to the Orange County maps. Looking at this, you might notice that the points are a lot more spread out than in Rensselaer County. What we found was some of these smaller towns and villages are actually detecting PFAS in their Finnet water, for example, the village of Florida. And now, again, these concentrations are within legal limits. They're things like 2.16 parts per trillion, but they are still seeing some detections. One cluster you might notice is the New Windsor area, pictured here. Like Hoosick Falls and Petersburg, they had a drinking water crisis. It's Newburgh and New Windsor are both home to about 20,000 people, and you can see the source of that contamination on this map here. The Stewart International Airport and the Stewart Air National Guard Base. On site, they used aqueous firefighting foam, whose main ingredient is PFAS, and during fire safety testing, they would periodically discharge it to make sure they could put out a fire, but in the process, over years of doing this, they contaminated the water supply. And this point here is the drinking water treatment plant for the city of Newburgh. You can see some of the concentrations they saw when they first identified they had an issue. And I'd like to point out these concentrations are from the effluent of the drinking water treatment plant. So this is finished water going into people's homes. As a result, they ended up switching over to the New York City water supply, which thus far has been non-detected for PFAS. And I think that's a good segue into another thing we looked at in some of these places, which is remediation measures. Places with drinking water contamination do have several options. Two of the more common ones are here, which are transitioning to new water sources. So like Newburgh transitioning to the New York City water source. Hoosick Falls is considering switching to either the Tominac Reservoir or some other well fields. And then granular activated carbon or GAC filtration is also a common choice. Here's a map we made of some water utilities in New York State that have GAC filtration. You might notice a lot of them are in Long Island. That sort of relates to the work that Harmon and Carolyn did last year. So one more thing we looked at was the demographic and socioeconomic characteristics of places with and without PFAS. We chose 12 places, not just in Rensselaer and Orange counties, but around the state that either have recorded PFAS or no recorded PFAS contamination. And for the places with contamination, we tried to restrict the list to places that had both detections in their drinking water and at least one site being actively remediated. We also tried to match population sizes between the two. So for example, Canandaigua City and the town of Colony have about 20 to 30,000 people, as do Newburgh and New Windsor, whereas Clarendon and Owasco have fewer than 10,000 people, as do Hoosick Falls and Medina. You might also notice the sample size is relatively small. That's in part because we were trying to match populations, and it's actually very hard to find places that have no PFAS contamination whatsoever. So we had a very small control group. So doing this, we looked at 20 different variables related to housing and socioeconomic and demographic characteristics like percent Asian, percent white, percent in poverty, home ownership, vacancy rates, amount of money spent on life insurance, things like that. And we found 3 variables that had somewhat significant relationships with PFAS. So percent in poverty and percent identifying as Hispanic were correlated slightly with more contamination, and percent home ownership, less home ownership was correlated with more contamination. So using all this together, we made a story map that combines some information from both last year and this year. One thing we didn't include was the statistical analysis work that I just mentioned. Because we did have such a small sample size and because equity is such an important issue, we thought the topic warranted some more research before being shared with the general public. But that's sort of the beauty of the story map. We're hoping in the future that more case studies and more data and more information, especially with the new MCL related data coming out soon, that this can be added to and will eventually give a very comprehensive view of New York State's PFAS contamination issues. So that is my presentation. Thank you, and questions. And one last thing is this a map I made for fun of PFAS in Dutchess County, just to show that PFAS is everywhere if you look for it.
Rasil, go ahead.
Thanks, Julia. That was great. Could you tell us about how effective GAC has been for the removal of PFAS?
That's something we looked into a little bit. So these utilities that have installed GAC have been testing to make sure that it's actually removing the PFAS, and they found that they're fairly effective. What we looked at, all of the PFAS that were detected were successfully removed. So anything in the effluent was had a zero concentration in the effluent for both Hoosick Falls, New Windsor, and the village of Garden City in Long Island, which were the three that we sort of used as examples.
Thanks.
T, you have a question?
Yeah, thank you so much for this presentation, Julia. It's kind of frightening. How do communities know if they have PFAS?
Yeah, that's a really good question. In some cases, communities won't necessarily know, in the sense that there are many places around the state that are being remediated for PFAS. So for example, if you have like a small manufacturing facility, you might not know that's nearby. But in terms of drinking water, if you do have a major contamination issue, the state will generally notify you and let you know that there is an issue and you should not drink your water. Sometimes that does, or in the past, that slipped through the cracks before New York State's MCL testing started happening, and they started requiring utilities to test for PFAS. Because, for example, in Hoosick Falls, it's a very small village. They weren't required to test. They weren't included in some of the monitoring programs like the NYSWRI, so they didn't know until they paid to get their own water tested.
Can I ask one question? Fantastic presentation. Thank you very much, as have all of the presentations been today. Congratulations to all of you. Julia, I wondered, because these are at fantastically low levels, 1 or 2 parts per trillion, did your study take into account any chance of contamination during sampling?
We generally didn't. We sort of took the values that like the water utilities and their annual water quality reports were reporting, sort of took those at face value and hoped that they were doing it correctly.
I have a quick question. Julia, I know this wasn't quite the focus of your research, but it seems to me like waiting for community to have high rates of cancer before things get dealt with is a real loss. In thinking about PFAS and maybe how these things might be regulated in the future, do you have any thoughts on like better approaches, how these regulations should come out to get ahead of the harm rather than waiting to address it once it happens?
That's a really good question. In general, emerging contaminants can be an issue because it's sort of on the burden of us and like the government and regulators to make sure that there is an issue and like a health hazard as opposed to companies that are making these products. In terms of PFAS, I'm really excited about the New York State MCL that's coming out. Water utilities will be testing all of them from the smallest to the largest. So that's nice. And the EPA has been promising to regulate PFAS or PFOA and PFOS at least for a while. So that's promising as well.
All right, thank you so much, Julia. That was awesome. We are going to take a quick break. Thank you guys for sticking with us. After our break, T and I will go. We'll be the last two. So if we could come back at 3:25, then I will start.
All right, all right, all right. We will begin at 3:25. And I will introduce you, Ashley, don't worry. All right, 3:25. Next up, we have Ashley, who is a Master's of Environmental Engineering in the BEE school. She will be talking about ROAs today, which are Riparian Opportunity Assessments. Are you ready, Ashley?
I sure am. All right, thank you so much, T. And you guys can see my presentation over there, full screen. Perfect. All right. Well, hi, everyone. I'm Ashley Sissel. I'll be presenting work on the improvement of the New York State Riparian Opportunities Assessment. Julia Gonzalez also worked on this project with me, and our mentor was Kristen Hitchka. So first, I want to introduce to you what a riparian area is. So it's the interface between the aquatic and upland habitats. So you can see here this beautiful interface between this river and what looks to be maybe a agricultural field. They provide with flood mitigation, water quality improvements, wildlife habitat, and ecosystem resiliency. Unfortunately, these areas have been a target for destruction, which causes a cascade of negative effects. When riparian areas are unhealthy, water quality declines, stream banks erode quickly, floodwaters normally held upstream are more quickly conveyed downstream, causing flooding in cities nearby those rivers and streams. Native aquatic wildlife, such as brook trout, are becoming locally extinct in their once native habitat. So New York State has a program to help landowners repair riparian buffers called Trees for Tribs. With the help from the DEC and the New York Natural Heritage Program, they created a tool called the New York State Riparian Opportunities Assessment, which I'll be calling the ROA from now on. The ROA has improvements that can be made, which I'll be addressing later in this presentation and throughout. Our task is to figure out which improvements will be best to solve those problems that users are having with the ROA by comparing it to existing technologies. We'll also interview users to get their experience on what needs to be improved. Then we will show a method of implementing the ROA for a very specific purpose. And then we also we performed that case study with an instructional guide, and we plan on giving those recommendations to the Trees for Tribs program. So for a more in-depth explanation of the ROA, it's called a suite of tools that assist in the identification and prioritization of streamside sites for restoration and protection. It uses what we'll call as indicators to quantify the condition of an area. So ecological significance, known water impairments, erosion index, all of those indicators, although they're all indicators of ecological health or ecological stress, which can be shown by this H and S in parentheses. Here's a look at the interface of the ROA. It's a GIS map that's divided into a size called a HUC12, colored to show the score of the indicators in that area. So dark blue, like over here in the Adirondacks, is signifying a high ecological health score. And then on the other side of the scale, we've got a dark red. So areas like New York City, Niagara Falls, Syracuse, and some of the Finger Lakes areas are signifying high ecological stress. So we wanted feedback from the people who use this tool and to gain insight based on their experience. We received responses from 13 users, developers, and administrators, all who work for places like the DEC, watershed groups, river coalitions, land conservancies, and the Department of Agriculture. Here's an accumulation of some of the insights that we found. There were a few purposes for the uses of the tool, such as outreach and grant development, quite a few known users of the tool, like landowners, municipalities, conservation groups, and even a teacher who used it in their classroom. We had lots of opinions on what the respondents thought the tool lacked, such as indicators that were defined to, well, it lacked indicators that defined at a smaller area. They wanted a tutorial for the tool, flooding indicators, and advocacy for the tool. So as we were receiving user feedback, we read through the documentation for 20 different riparian tools, looking for the similarities on the indicators that they use. So there were a total of 115 unique indicators, 20 of which were used in the ROA. Others varied between like 3 and 26 indicators per tool. And some indicators were also shared between the tools. So for instance, the Vermont Wetlands Inventory Map and the ROA both used ecological significance as an indicator. Here on the right, we have the 20 tools that we evaluated, but you don't need to remember these. It's just an idea to get how many we looked at. And if you want to come back and discuss it further, we definitely can. So first we want to look at the ROA and the number of tools that share indicators with it. So here we can see the indicators on the y-axis, and then the number of tools that use the indicator on the x-axis. So we have quite a few that are unique to just the ROA, like the landscape condition assessment and matrix forest blocks, though we do have a couple that are popular with other tools, like impervious surface area and known water impairments. Those are used by 6 other tools. And to get a perspective on what those unique indicators mean for the tool in general, we compared the number of unique indicators for the ROA to that of other tools. So the ROA has the highest proportion of unique indicators at about 70%. That unique indicators are that orange bar, and that blue is total indicators. So it has about 70% unique indicators. So if we're using the popularity of the indicators as a metric on which indicators the ROA should include, we wanted to find the overall most popular indicators. The ROA's 2 most popular were also 2 of the most popular overall, this known water impairments and impervious surface. Plus there were 4 others that were popular. Current historical land use was used by half the tools. Hydric soils, habitat type and quality, those were all tied with 8. So as mentioned in the interview section, users of the ROA suggested that a flooding indicator would be useful. So to investigate this further, we used the 5 prioritization tools out of the 20 that I listed earlier that did have flooding indicators, plus 15 new tools that were ecosystem services tool that also had flooding indicators. We found 76 total indicators in this category. And as you can see in this graph, we also have a higher number of unique flooding indicators in this group. So what we did was we categorized each one of those indicators into 3 types of flooding. There was attenuation, runoff, and retention, just to get an idea of what types of indicators were most popular. So there were 33 retention, 31 runoff, and 12 attenuation indicators. So although there weren't flooding indicators that were shared by a large number of groups, if we were to recommend a specific indicator to add to the ROA, here are a couple of the most popular indicators that we think would be good candidates. And although the ROA doesn't specifically outline a flooding indicator, some of the indicators that were used in the ROA were also used in other tools as parts of their flooding indicators. So if we can combine all of these in the ROA, we can create a pseudo flooding indicator method of evaluating riparian areas that would provide the most flood mitigation benefits. So this would be useful to users that need to evaluate flood risk with the tool the way it is before any improvements could be made. So we chose a location in the Wallkill watershed to overlay these 5 indicators in the ROA. So darker gray indicates less flood mitigation benefits, and lighter gray would be those areas that could be prioritized for riparian restoration. So in conclusion, we recommend a few ways to improve the ROA. First, we need improvements on the increasing the user base by evaluating the indicators at a smaller area. We needed to add additional popular indicators such as the current historical land use, hydric soils, habitat soil and quality, restoration potential, and a retention flooding indicator. We think a specific advocate for the tool is necessary for the growth in new users and continued use by existing users. Plus an update in the data would be useful as the tool ages, and website improvements to more easily find and watch the tutorial will allow new users to more easily learn the nuances of such an advanced tool. So with all these improvements, riparian restoration efforts and funding in New York State could be potentially optimized, and hopefully methodologies for choosing optimal restoration areas can be further standardized. So that is the end of my presentation, and I will now take questions.
Excellent presentation.
Thank you.
Shannon, do you have a question?
Yes, hi, Ashley. Thank you so much for the presentation. I'm so glad that you decided to focus on this. I'm with our DEC's Great Lakes program, and we were actually part of the first pilot phase of this project, developing it initially for the Great Lakes Basin, and then working with Division of Lands and Forests to extend it throughout the state. And you know, I just really appreciate a lot of the feedback and analysis that you were able to do, and I'd love to get a copy of this so that we can kind of take that and really evaluate how we might be able to kind of make some of these improvements. When we first developed the tool, we of course intended to go in, you know, every 5 years or so and really make sure that some of these updates and customizations were kind of continuously improving it. But in any case, this is a great reminder. I had a question though regarding advocate for the tool, and you know, again, I agree. Did you were you able to kind of spend much time in thinking about, you know, what the best ways to do that were based on, you know, the user groups that you were able to reach out to during your project?
You know, we hadn't considered a lot of like who might be the best position for the for that kind of role. We got to think about it a little bit. So someone like the state coordinator. So for everyone else, the Trees for Tribs program is set up with like a state coordinator, and then there are regional coordinators who also, you know, do smaller scale riparian restoration. And so we thought that the state coordinator would be a good position for that. That advocacy for the tool, or maybe someone who uses the tool frequently with a lot of experience in it, and potentially would have time to gather up that information and be able to give it to whoever can make those improvements.
Yeah, and just curious also along those lines, were you, did you have, I guess, did you find that any soil and water conservation districts were using the tool in your project?
We didn't get to talk to anyone from the soil and water conservation districts. The people that we did get to talk to, they said that they knew of soil and water conservation districts that did use it.
Oh, great. Cool. Thanks so much.
Brian gets the last question with, does anyone use the tool to evaluate capital T, lowercase f, capital T, or other riparian restoration efforts?
Does anyone use the tool to evaluate? They do, yes. So that was one of the main goals of the Trees for Tribs ROA. So yes, we did talk to people who did use it for that. We would like to extend that user base to a lot more people who are using it for riparian restoration, including, you know, more homeowners or more landowners, more watershed groups, that kind of thing. So yeah, we did get to know people who were using it for Trees for Tribs, but like I said, we would like to extend that user base to many more people.
Check out the comment from Lydia Brinkley.
Ooh, awesome. Yeah, 22 soil and water conservation districts. Awesome.
We do have actually one more question. Time for one more question. Do you think there are any improvements that you could do for the ROA, Ashley, that you personally going through these 10 weeks of research?
Yeah, so honestly, I think that some of the best, maybe like if you're prioritizing the prioritization tools, I think one of the fastest, easiest ways to improve this tool would be those website improvements. So there is a tutorial for the tool, although it's not intuitive on how to use it. And you also have to download another program to actually go and watch it. So I think it would be ideal for someone who's just checking out the ROA. They'd be like, oh my gosh, this is intimidating. How on earth do I use it? There's a box right there. It says, click the play button and you can see how to use this. So I think that would be a very quick and cheap and easy way to improve this interface.
Awesome. Thank you so much.
All right, and since that concludes my project, it would be my pleasure to introduce our very last presenter, the anchor leg of WRI's webinar today. And coming in on the home stretch is T. He's a master's student, or they're a master's student in regional planning with concentrations in community and climate change resiliency, planning. So T, the floor is all yours.
I will take the floor. Excellent. All right, let me share my screen. Hi, I'm T. Today, I will be presenting about barriers to a specific flood mitigation program called the Community Rating System. And what I hope to do is to take these barriers and then talk about how we can approach climate change adaptation governance in New York State. So I'm going to start out kind of macro scale, locate everything within climate change, then hone in on the importance of flood mitigation as a way of kind of assessing how climate change governance is working through a specific federal program called the Community Rating System. I'll talk about costs and benefits, and then the barriers, and then I'll end on some recommendations. So this is important because by looking at this one specific program in flood mitigation, we were able to better understand the greater climate change adaptive capacity of communities in New York State. Set this up, all predictions say climate change is real and climate change is happening. It doesn't matter if you look at the increased temperatures that are being predicted, high and rising sea level change at New York City, or what I think is most interesting is that the intensity, which is this red line, and the frequency of storms of a 100-year flood are gonna actually end up being something more like an 80-year flood. And this last graph is really important because if you look over the last 10 years, from 2011 to 2019, flood specifically has cost the state of New York $33.3 billion, which is a really great way of kind of focusing in on it because it is the most deadliest, the most frequent, and the costliest natural disaster there is. And so if we can focus on the worst parts of climate change, we can then kind of expand out and focus on like rising temperature and other type of things. And because flooding is directly connected to climate change. So let's do this. To understand, it's kind of necessary to take a step back and look at the history of flood mitigation in the US. And this is a timeline. As we can see, essentially, the US has moved from these hard infrastructure practices of building dams and levees to kind of more focusing on flood insurance as a way of mitigating change. And in 1990, they started this new program that I'll be talking about, the CRS. It hasn't really worked, as Joseph kind of mentioned, and that has to do with actuarial risk. And to kind of visualize why the National Flood Insurance Program, NFIP, isn't working, nothing better than to look at this graph, where we see that how many people are paying in, the flood premiums that they're paying are not matching the costs that are coming out of it. And Katrina, Sandy, and Harvey have really shocked the system. So the community rating system is a really interesting way of approaching that. Also looking at this in another way, debt is skyrocketing because of flood insurance, flood claims. So with that, let's talk about this one specific flood mitigation program, the CRS. The CRS has a very basic premise. It's a voluntary financial incentive system. So if a community or a municipality goes in and does flood mitigation activities, they get points, and these points then help them work up this tiered system so that the citizens specifically can get discounts on their flood insurance premiums. So the more a municipality does to protect itself from a flood, it is rewarded by not having to pay as much flood insurance. The premise is pretty brilliant. It seems like everyone wins, which begs the question, why is participation in New York State so low? It's 2.8%. For comparison, the largest one, Florida, is at 64.2%, even though they've experienced similar amount of flooding events in the last 10 years. So what is the difference? Why is there such a big difference? To kind of understand that, we have to take a closer look at how the CRS works. It's essentially 19 flood mitigation activities that are broken up into 4 categories, or what they call series, that include public information, mapping a flood zone, relocating people out of a flood zone, or putting stream gauges or river gauges to make sure that you can prepare for a flood. To visualize what these activities look like, the big 3 are freeboard requirements, which is raising the first floor of a house above the flood zone area, so 3 feet in this example. Mapping flood zones, so kind of understanding where the flood, if a flood was to overcome a bank of a river or stream, where is it going to impact? And also building infrastructure that allows wet flood proofing, essentially allowing water to go into the first floor and stay there for a while without having any structural damage to the building. It gets a little bit more complicated because you would think that if you just did the activity, you get the points, but there's an actual process to the system. And this kind of goes, each activity has an element, and these elements are then adjusted by the impact. Either the impact of the flood zone, the special hazard flood area, or county growth adjustments. So if a county decreases in population, a participating community could lose points. All these are calculated, and then all of these activities are totaled up to give the actual classification, which gives all the citizens their flood premiums. So let's just do an example. The example would be that a town has 100 buildings in the special flood hazard area, but they can only collect elevation certificates of 25 buildings. And elevation certificates are what every community has to do. So you would do elevation certificates divided by the total amount of buildings to get 25%, and then you'd multiply that by the actual activity. And for elevation certificates, that's 116 points. So in total, you've just done 29 points worth of work, and you have to get above a 500 to participate. So you can kind of see how this is very, very complicated. So also with that, let's just take a step back and understand CRS is voluntary, it's cost effective. So communities opt in. Communities can opt in as much as they want, and they will get the points in return, but varying on the actual scale. If we have 2 communities with the same population, but one has a bigger flood zone, then that one might get more points than another community. So it's very, very varied. Barriers. Barriers are broken down into 2 things, particularly in New York State. The first one is that there's a lack of resources to do this program. And the second is that there's a regulatory mismatch. And this regulatory mismatch has to do with New York being a home rule state, which essentially means that land use law is not explicitly stated in the state constitution. So it's given to all 1,550 New York State communities, 1,550 opportunities to do whatever they want with land use authority. And what that also means is that there are 1,550 communities. All these communities are very small, so staff is an issue. Some staff double up as the town clerk and building codes coordinator. That also means time. The burden disclosure for this program is 45 hours per response. And if there are 19 activities that involve different types of responses, you can see how this time adds up. Also, the building codes department may not talk with Parks and Rec. And then we have this data management problem of all these documents are being passed around and no one actually knows what the elevation certificates are or the documents that you need. And CRS is like many federal programs, you have to prove that what you're doing with paper, and you may be doing all the right things, but if you don't have the paper trail, you get no points. Which is also why GIS analysis, a very technical and expensive program, all Series 400 points are actually GIS, spatially understanding the risk of the flood zone. And if you don't have that software, then you are not going to be able to get those points. Zoning is also an issue for a small community where, you know, what property is the most valuable in a town? Waterfront property. So a town has to decide, do we preserve this waterfront property to make sure that we're prepared for a flood, or do we allow businesses to build on it so that we can collect property taxes and then build our revenue and provide our basic services? That also being said, building codes is another regulatory mismatch where New York does not have standardized building codes across everything. They're kind of varied by different parts, and then towns adopt these building codes. So honing in specifically on building codes, if you relook at how CRS allocates points, you're going to understand that land use authority and building codes are the majority of how communities can get points. And if you have power, but then you've got this difficulty of deciding whether to protect yourself or to make money, then it sounds like the community is not going to be able to do that. On top of that, for energy code appliances, there are different climate zones. So New York has different building codes in different areas. The CRS has a universal building code that other states adopt, but New York has not. And therefore, New York is not able to participate at the same level that other states are. With all this said, I would recommend that we consolidate building codes at the state level, that we either give regions or counties support with GIS analysis so that all the Series 400 are housed in a specific regional area instead of having to put that cost and burden onto small communities, as well as documents and data management. But this presentation doesn't end just with here. I'm hoping to do in the future a statistical analysis comparison between Florida, New York, Pennsylvania, because there's this perception that CRS is mostly for richer communities. And in some ways, you could actually see how that actually works because communities with more policies are more likely to participate. Florida has the most national flood insurance policies in the entire country. But I think it's also interesting because it may not actually be like that. Social equity is important in the sense that Syracuse, most of the population is low income, whereas something like the village of Piermont is mostly lakeside houses that have seasonal tourists. So understanding how this works with CRS is important to then kind of think about climate change in a greater sense. And because it costs so much to get through all this, consulting fees and trying to figure out how this program works, most towns then outsource this to a consultant. I'd like to be able to try to generate an algorithm to see if they have the local capacity to actually participate instead of having to spend that money to then realize, actually, we don't have the capacity to do this right now. And yeah, that is everything. Are there any questions?
Yeah, T, I have a question. So you were talking about all of those barriers to becoming a part of the CRS for New York State. And so I was wondering, like, for the places that don't have such a low participation rate, they like Florida and Texas, did they overcome those barriers, or did they just not have those barriers at all in general, or, and what can we learn from those states?
So Florida specifically took a very interesting approach by locating, well, they did a CRS initiative in 2015, 2017, and they housed everything in the Bureau of Mitigation, which was in the Florida Department of Emergency Management. And they send out teams to communities to then help the communities kind of compile the checklist, and there's a checklist to apply to this program. So they would help with the very basic stuff. And by having this support coming from the state level down to the communities, they're able to increase the participation rate in those 2 years. And now it is standardized. It was initially a grant application, but now it's built in to the actual state government.
Very cool. Raywa, did you have a question?
You asked my question, Ashley.
Nice.
I wanted to ask, how did Florida overcome these barriers?
We also have a question in the chat from Brian. Did we ever understand whether counties can be CRS certified in New York?
There was a directive that said counties could not participate, but according to the 2020 addendum, it says that not only can counties participate, but nonprofits, or other type of organizations. So you can go on behalf of a town now. Yeah. And so you may be asking, well, okay, that was so state level. What can I do? I got the answer for you. So what you can do to move New York State climate adaptation or wherever you're at is actually reading your community zoning ordinances. A really good example is the Village of Piermont has a building codes, as every community does. But if you look closely, you'll see that there's a maximum building height that says 35 feet. And in this part of the town, most buildings are around 32, 33 feet. But if they're in the flood zone and they want to raise them, their house as freeboard standards are, they would then need a variance to then do this because they're trying to preserve the viewscape. So understanding your own community and seeing that there are height restrictions, and then that's going to prevent people from mitigating flood activities is one way to help your own community move forward.
Very cool. Does anyone have any other questions? All right. Well, if that concludes T's presentation, then that would also conclude our webinar for WRI. Thank you all so much for joining us. We appreciate it. Friends, family members, mentors, WRI in particular. We appreciate your support so much, and thank you for sticking with us through this whole time. We are, like I said before, we are recording. So if you were interested in getting the recording, you can always reach out to WRI and we can provide that to you. And yeah, that'll conclude our webinar. Thank you all so much for joining us.
Good job, everybody. Really well done.
Did anyone from WRI have comments or final wrap-ups?
Not to outdo you, Ashley, but I just wanted to say great job to everybody. And, another 10 weeks, and I think you would have all totally solved these problems. But just acknowledging everybody, very professional presentations, good results, and all in 10 weeks over, what was a logistically challenging summer. So congratulations, take a break, go enjoy yourself. You've all earned it. So thank you.
Absolutely. All right, and with that, I will stop recording and